more about technology. Right. Let me just have a look. Okay, I hope you can all see what is on the screen. If you cannot see what's on the screen, then Mr. Farmer can maybe indicate that to me. Otherwise, I'm just going to continue. So, firstly, when you go into an exam, it's very, very important that you know what you will be writing on in paper 1 and what you need to study for paper 2. So I'm just going to go over this quickly, because also what is very useful is on the right-hand side over here, it also tells you how many marks each section counting in the final exam. So you will see, for example, that meiosis, which hopefully we will be able to cover today, is dealt with in paper 1, and it's about 11 marks, but then it is also in paper 2, where it's about 12 marks. Then in paper 1, we're going to be covering reproduction, so human reproduction and all the other reproductive strategies in other animals. Then we do responding to the environment, so that would be your nervous system and the eye and the ear, the human endocrine system, homeostasis, which is an important section, which we also did in grade 11, responding to the environment, but now we're talking about the plants, and then human impact in the environment, which you also did in grade 11. Then you'll see in paper 2, we're going to be covering DNA, which counts 27 marks, so that's quite a lot. Meiosis I've spoken about already. Genetics I want to do in detail with you in the next tutoring lesson, so that counts nearly 1/3 of paper 2, and then evolution, which also is a very big section as well. So I find this is very useful, so just bear that in mind, so that you definitely, when you go into the exam, you are well prepared and you know exactly what you're going to be writing on. I'm going to share my screen and show you something else. By the way, this is in Mind the Gap, and you should also find this at the front of your textbooks, I'm sure, and Mind the Gap is one of the e-learning resources that the Western Cape Education Department has provided, so all of you should have access to this. Right, then the next thing I'm showing you is the examination guidelines. And these, I always say to my matrics, they need to refer to all the time when they make their notes and so on. Because these guidelines tell you exactly what you need to study. Because everybody has got different textbooks and cover different sections. But if you follow these examination guidelines, then you will know exactly what to study. So you can see DNA, I may as well show you this today since we are covering this today. So firstly, we do the revision of the structure of the cell with an emphasis on the ribosome, cytoplasm, and the parts of the nucleus. So you would have also done this in grade 10 when we did cell structure. Then we need to know that nucleic acids are made up of nucleotides. Those are the building blocks of nucleic acids. You need to know about the 2 types of nucleic acids, DNA and RNA. Where is DNA found? So you need to know about the nuclear DNA and the mitochondrial DNA. A little bit about the history of the discovery of the DNA molecule. And then these scientists or people in particular you need to know about: Watson, Crick, Franklin, and Wilkins. The 3 components of a DNA nucleotide, here they are bulleted. The natural shape of the DNA molecule is a double helix. A little diagram of the DNA molecule you should be able to do to illustrate its structure, just a simple diagram. The functions of DNA, so we'll talk about this later, but that it carries hereditary information, and that's very important for protein synthesis. Then we're going to talk about the process of replication, where the DNA makes a copy of itself, and then the definition of a DNA profile, uses of DNA profiling, and then we speak about RNA as well. Where is it found? What does it do? what is its structure, and a little diagram of messenger RNA and transfer RNA, and then this section on protein synthesis. And you will see what is quite nice, because sometimes one doesn't know exactly what to summarize in your textbook, but here in your examination guidelines, transcription has been explained step by step, and so is the process of translation being explained step by step. So these examination guidelines I find are very useful. Okay, then we're going to go on to your tutor guide, which I hope all of you have got in front of you, but I'm also going to be sharing it on my screen over here. There we go. Okay, now some of this, many of you will know this information already, so I am going to go quite quickly, but over here we want to know, when you walk into the exam, how are you going to be answering your paper? So firstly, remember you've got 2.5 hours for each of these papers, and from experience that is really, really plenty of time. So the first suggestion I would make is do not rush your paper. You will have enough time to complete it. Read your questions very, very carefully. All the questions. Read them carefully once, 2 times, make sure you understand it. Also, you can obviously use your question paper to make little notes, to circle things, to make little ticks and so on. And remember, especially in a lot of the questions, a lot of information is given to you in the question already. So that is why it is so very, very important to read the questions very, very carefully. Now, in section A, which counts for 50 marks, you will see it always starts off with multiple choice questions. And people often think, oh, multiple choice questions, that's easy, but it isn't. You really need to know your work very, very well to answer these multiple choice questions. And then before we go over how to answer these questions, I also want to ask you, matrics, please, when you write down, because you're only going to be writing down a letter, A, B, C, or D. Make sure that the examiner can see that it is a D and not a B. And do not make letters that look like they could be a B or they could be a D. So also, if you go over the paper and you realize, oh, you've made a mistake and you want to correct that question, don't go over that letter and do it in ink and expect the examiner to work out what that letter is. Simply cross it out and rewrite it, okay? You should never be allowed to use Tipp-Ex. It's much better to just simply cross it out and to rewrite the answer again. Okay, so let's have a look at some of these examples of these multiple choice questions, which you're going to be answering. Remember, they are 2 marks each, so that's quite a lot. So you need to be very careful when answering them. So with multiple choice questions, you're going to see there's first going to be a statement. And what I suggest you do is you do not even look at the answers. Just look at the statement and try and answer it without looking at the answers. So over here, let's just quickly go over it. So there are 4 possible answers. I'm over here. If you see your answer, circle the letter. Look at the other possible options to check that none of the other options is a better option than the one you have chosen. If you do not see your possible answer, then cross out the 3 options that you think are wrong. And then also don't leave a multiple choice question unanswered. Okay, so here's an example. It says here, the part of the brain that receives nerve impulses from the semicircular canals is the. Now, don't look at the answers. Now, you just look at that first part of the question and just go back to what you've studied on the hearing and on the brain and so on. So you know that the semicircular canals, they are involved in balance, right? And then when you studied the brain, you had to study the cerebrum, the cerebellum, the corpus callosum, and the medulla oblongata. And then you ask yourself, right, which section of the brain was responsible for balance? The part of it that receives nerve impulses from the, and then the answer we know is the cerebellum, all right? So we haven't, and there we see the answer is there, so we put a little circle around it, but then we just, because we're not 100% sure perhaps, we just look at the other possibilities. So cerebrum, is for higher mental functions, for reasoning and logical thought, and it's the seat of all sensations. The cerebrum is involved in hearing and sight and so on. And there you can see the hypothalamus is responsible for homeostatic control mechanisms like thermoregulation, the dilation and the constriction of your blood vessels on a hot or a cold day. And then the medulla oblongata controls all your autonomic reactions, like heartbeat and also the rate and depth of breathing when your carbon dioxide levels increase. So now we've ruled out the other answers. So yes, cerebellum is correct. And then in your answer book, you write B. You don't write cerebellum, you write B. Okay, let's do one more example. The next question says, which one of the following is true about genes? And then we think of our section on genetics and DNA, and we remembered a gene is a segment of a DNA molecule which codes for a particular characteristic. Right, so now we look at the answer. Oh, A, there it is, codes for a particular characteristic. So we circle that on our question paper, but just to make sure it is right, we quickly look at the other alternatives, and then they also have made up of amino acids, but we know proteins are made up of amino acids. Made up of RNA, genes are not made up of RNA, they are made up of DNA. Found in a ribosome, no, genes are not found in a ribosome, they, as we said, they make up the DNA molecule. So the A is correct, and we write down A on our exam paper. Right, so those are the multiple choice questions. As I said, they are 2 marks each, so be very careful when answering them and don't just answer the first thing that comes to mind. Now the next type of question that you get are these matching columns, all right? So let's just read through this together. This question type is found in section A as well, and these are also 2 marks each, so that's a lot. Normally, a key is provided to be used to present your answer to the question. Each question starts with a description or statement. 2 possible answers are provided. When answering this question, read the description and each possible answer separately. If the possible answer is correct, make a little tick next to the possible answer. If the possible answer is incorrect, make an X next to the possible answer. And then use the key to present your answer. Now this key, there we go, is sometimes a little bit confusing to people, but let's just read what it says. So you, in your answer paper, you are only going to be writing, you're either going to write A only over here, or you're going to write B only, or you're going to write both A and B, or you're going to write none. So that's what you're going to be writing down as one of your answers, right? So there it says A only, B only, both A and B, or none. All right, so let's look at the example we've got here. It says here, a young bird is helpless soon after hatching. Now again, I would quickly just think back on what we've studied in that chapter on reproductive strategies. And then I remember, oh yes, a helpless individual needs lots of, the parents need to look after it, they need to feed it, they need to keep it warm, and so on. And these terms are always very, very important in life sciences. You need to know exactly what certain words mean. So we go over this and we say a young bird is helpless soon after hatching. Is that precocial development? Now, precocious is often, in the English language, is often if people are actually mature quickly for their age, right? So precocious individuals are easily able to look after themselves, right? So precocial, no, that's not going to be correct. Altricial, when we've learned our life sciences, we know is where the young need lots of care from the parents and need to be fed and so on. So the correct answer here is altricial development. But now, as I said, we're not going to write down the word altricial development. No, we're going to say the correct answer is B only. And that is what we're going to write down. All right? So let's do one more example. Here it says a sex-linked genetic disorder. All right? So you studied these in your chapter on genetics, right? And these are often carried on, they are often recessive alleles on the X chromosome. So the conditions are often more prevalent in males than in females because, of course, males have an X and a Y chromosome, whereas females have two X chromosomes. Right. Is hemophilia, remember, is when the blood doesn't clot. Is that a sex-linked genetic disorder? Yes, it is. Color blindness is far more common in males. In fact, I think 1 in 12 males is colorblind. So that is really quite a lot. So this is also a sex-linked genetic disorder. So both of these answers are correct. So then we would write down both A and B. Okay, so those are two important types of questions. Now, that's now the short questions. Now we're going to go on to a section on drawing of a graph. So we're now on page 4 in your notes. I hope you're all following. Now, nearly every question paper that I've seen, at some point you need to draw a graph. And this is a skill that you've probably learned already in grade 8. So every year in life sciences, the learners are expected to draw a graph. And remember, you get different types of graphs. You've got a line graph, a bar graph over here, a histogram, and a pie chart. Now, firstly, if you need to draw a graph, you need to have the right equipment. So what do you need? You definitely need a ruler, you need a pencil, you need an eraser, and then for a pie chart, most definitely, you need a compass to draw your circle, and you need a protractor because you're going to be measuring angles. And then very, very important, you need a calculator because sometimes it's quite difficult to measure those, to work out those angles. So please, when you go into the exam, you make sure you've got all the right stationery with all that equipment with you. Then, whenever you draw a graph, you need a heading. And this is so important because your heading mustn't be vague. In your heading, you are going to explain what that graph is about, exactly what that graph is about. And you have to include your independent variable and your dependent variable. So, for example, you could have a graph, for example, the effect of altitude on the number of red blood cells. So there we have included our independent and our dependent variable. Just a little bit of a revision. So the independent variable refers to the factor that is being investigated. And this factor is often manipulated or changed by the investigator at the start or during the course of the investigation. So in my previous example, the independent variable would be the altitude. Altitude, of course, is height above sea level. And we know that as the altitude increases, so the number of red blood cells increases as well. Because, of course, there's far less oxygen with an increase in altitude. Then your independent variable is always plotted on the x-axis. The x-axis is the horizontal axis. So make sure that you do that correctly because one can get lots and lots of marks when drawing a graph. But of course, the graph has to be correct. Then your dependent variable refers to the effect of the independent variable, but it is usually the one that you measure or you observe. And you always plot this one on the y-axis of the graph, which is the vertical axis of the graph. So in my previous example, your dependent variable, what are you measuring? We're measuring the number of red blood cells in the blood, right? So that is the dependent variable. Then you're going to label your x and y axis, and you get a mark for doing that. And then, very important, you're going to have to include the units. So for example, in my previous example, altitude would probably be measured in meters, and the number of red blood cells is probably going to be measured in cubic millimeters or something like that. So these must be included as well. So other units would be degrees Celsius, seconds, years, number of organisms, etc. So do remember to always include the unit. Then work out an appropriate scale for the axis. Now I'm just going to quickly go on and show you this one graph over here. So when we talk about the scale, we're referring to these intervals between your points over here. Now, what lots of learners do, they make this mistake, is that they simply look at the table and they look at all the numbers or the data that's being given on the table, and they fill in that data on the x and the y axis. But now one must not do this, all right? So the scale means that these intervals between the values need to be exactly the same. So you can see in this graph, the intervals are tens. So here we are going up in tens. 10, 20, 30, 40, 50, and so on. Here on the y-axis, the intervals are 200, because here we're going up in 200s. 200, 400, 600, 800. And this is very, very important that you get the scale correct. Now, I hope you've got a pen or a pencil in your hand and a piece of paper, because what I want you to do very, very quickly is, actually before you do this, is normally what I do if I have to work out the scale is I look at the lowest value and then I look at the highest value in my table of data, and then I am going to work out an appropriate scale. So for example, if my lowest value is 0 and my highest value is 100, then I'm not going to go 0, 2, 4, 6, 8, 10, 12, 14, all the way till we get to 100, because then you're going to have lots and lots and lots of points that you've plotted on your x or your y axis. In that case, an appropriate scale would be to go up in tens, 0, 10, 20, 30. Now, what I want you to do is, on your piece of paper, I want you to work out a scale. If the lowest number, for example, is, let's make it 10, and the highest number is 1,000, what would then an appropriate scale be? And then I'm going to give you another example. Let's say the lowest number is 0 and the highest number is, let's just make it 1. What would an appropriate scale be? So please just think about that quickly, and then I'm going to ask you in a minute, or I can't really ask you, but I'm going to tell you what the answers are. Okay? So the first one is, if you've got a piece of paper and a pencil in front of you, the lowest number is 10 and the highest number is 1,000. And then the second example is where the lowest number is 0 and the highest number is 1. Okay, I hope you've all thought about that. So in the first example, the logical scale would be to go up in hundreds. So you would go 0, 100, 200, 300, till you get to 1,000. In the second example, one would go up, I would probably go up by 0,1. So I would go 0, 0,1, 0,2, 0,3, 0,4, and so on. If my lowest number was, say, 0, and the highest number was 2, then I would probably go up by 0,2. So I'd go 0,2, 0,4, 0,6, and so on. So I do hope you understand that. That is very, very important that you get the scale right. So like I said, look at the lowest number, then look at the highest, and then write down what this, then obviously write those down. Now let's go back to what we were saying. So work out an appropriate scale, so that we've done. When drawing a bar graph, remember that the bar should be of equal width. I'll show you this in a minute. And the spaces between the bars should also be the same. When drawing a pie chart, you must use a compass to draw the circle of the chart and a protractor to measure the angles in degrees of the different sectors. Right, now sometimes you don't need to draw a graph, but there is a graph on your question paper and you've now got to interpret the graph. So always look, the caption, that's the heading of the graph. So look at the heading and read it carefully. And in that heading, it will tell you what the two variables are that are under investigation. And immediately write down which is the independent variable and which is the dependent variable. Also, if you look at the graph, you will see that the independent variable is always on the x-axis and the dependent variable will be on the y-axis. And then also, a line graph will show you the relationship between the two variables. So going back to this graph over here, let's first look at the heading. So the heading says, the graph below shows the relationship between the production of growth hormone and age. So those are the two variables. So you can see age is the dependent variable and it is measured in years. And the amount of growth hormone is the, sorry, I made a mistake there. Age is the independent variable. It's here on the x-axis and it's measured in years. And the amount of growth hormone is the dependent variable. And here you can see it's measured in microgram and it's on the y-axis. Now, if you were to, now often when you see a graph like this, you've got to write down what is the relationship between these two variables. Now, a mistake that the learners often make, and they get a bit confused because they've taught this in mathematical sciences, is they say this is a directly proportional relationship. Now, we do not want you to use those words directly proportional. If you look at this graph, you can see in a young person, so a 10-year-old, the amount of growth hormone is quite high, 200 milligram. Then when a person reaches 20, now you can remember most, you're going to grow very rapidly between the time you're born and 10 and even 15, but then the amount of growth hormone is going to decrease with age. So the relationship over here you can see is that the amount of growth hormone is going to be decreasing as the person gets older or decreasing with age. So when you write down the relationship between these two variables, always mention the independent and the dependent variable. Here we can see the trend on the right-hand side over here. As age increases, the amount of growth hormone secreted decreases. So just remember, don't use those words directly proportional. Okay. Right, then sometimes they ask you to read values from the graph. So for example, how much of the growth hormone is secreted at 30 years? So what you do is you get your ruler and you put your ruler up vertically over here on 30, and then where it touches the graph, you draw another line going across, and then you read the value over here from the y-axis. So that's the first one. And yeah, so that is also an important skill. So as I said, you take your ruler till it touches the graph, and then you read your answer off from either the X or the Y axis, depending what the question is. Right, here we've got a nice bar graph. So the bar graph is of the cranial capacity of different primates or species. And over here we've got all the different primates. These are all the genus and species names over here. And here we've got their cranial capacity, which is measured in cubic centimeters. There you can see the scale on the y-axis. They're going up in 100s, so the intervals are the same. And very important, you can see that this bar over here is exactly the same bar width as the bar for the gorilla, and that's the same as the width of the bar for Australopithecus, and so on. And also you can see that the little spaces between the bars are exactly the same. So this is a good graph, and you actually do get marks for drawing this correctly, showing that and indicating that the widths of the bars are the same and that the spaces between the bars are the same. And there you've got the unit, so this, and there's the label. Please don't forget to put a general label down for the x-axis. So it's not just good enough to write down the little labels for each and every single bar. You still have to have a general label, because what is this of? Okay, it is of different primates or different species. Okay. Then in the Mind the Gap, which, as I said, is available. Some of you maybe are using Mind the Gap for studying, or you've got the notes, and it is extremely useful to use. But it is, like I said, also available as an e-learning resource. But there they also explain to you in detail how to draw the pie chart and how to convert percentages into degrees. And there are lots of examples if you look at the old examination papers, which are also available on the Western Cape Education Department website. Do practice at least 2 or 3 of these pie charts, because these, as I said, you can easily get a lot of marks just for drawing the graph correctly. Right. Then we go on to calculations. Now, remember, life sciences is a science, and so you do need to be able to do simple calculations. So you need to be able to, for example, to work out an average. Very important when you do calculations, and I'm going to highlight it, you must include the unit in your answer as well. People often forget to put the unit. So they'll write, for example, if the answer is 30, it should actually be 30 cubic centimeters or 30 milliliters or whatever it is. So do remember to always put the unit in your answer. And then, of course, you need a calculator. So don't forget a calculator, and it can even be a simple little calculator that you need to be able, you don't want to lose marks because you couldn't do that calculation. Right, to calculate an average, I'm absolutely sure that all of you know how to do that, and it is very important. In this table over here, they've got a number of participants, 5 participants, and then they've measured the pulse rate in beats per minute and of these 5 different people. Now they want to know what the average is, the average pulse rate is. So what you've got to do is you've got to add these numbers, 76 plus 91 plus 95 plus 89 plus 89, and you're going to add those up, get your answer, and then divide it by the number of participants. And remember to show, or sometimes you also need to have a formula. All right, and that's also important. So write down the formula, excuse me, and then you write down, you fill in all the numbers and so on, and then you write down your answer plus the unit, because something like this could be worth about 3 marks for your formula, for your substitution and showing what you've done, because now we know that you understand this, and then for your answer and with the correct unit. So this is a skill that is also important in life sciences. Sometimes you have to calculate a percentage. I'm going to quickly have a drink of water, 1 second. Right, so here you've got a table. Again, now we've got altitude in meters and number of plant species. So here it says calculate the percentage of plant species found at an altitude of 1,500 meters. So we can't just write 60. We want to know what percentage of the total is found at a height or altitude of 1,500 meters. So there again, we've got a little formula over here. So it's number of plant species at 1,500 meters. So that's 60. And then of the total, so the total is 180. And you multiply that by 100 to convert it to a percentage. And there we go, there we've substituted correctly, and our answer is then 33.33%. So this is also something that you should be able to do. Then we go on to comprehension. Now, comprehension is similar to in English, I guess, when you do a comprehension exercise. So we are testing your understanding of we normally give you a passage to read. Could be a passage from a scientific journal or from a newspaper, and then we ask you questions on this to see whether you really understand it. Now, it's also, just remember that a lot of the answers to the questions will be in that passage. So read it very carefully. Read it 1, 2, 3 times and underline the important information to make sure that you understand everything well. All right? I don't think I'm going to do this exercise with you, but here you can see they got a little article all about diabetes. Now, we discussed diabetes when we did the endocrine system, right? So when we spoke about insulin and glucagon, the 2 hormones that control the glucose levels in the blood. So we discussed this over when we did the endocrine system, and that's quite important. And you can see this little article has come out of the source is diabetes research. So you read through it very carefully, and you underline important sections, make sure you understand it, and then you answer it. And as I said, the first few answers are normally in the little extract that they've given you, right? And then when you get to the other answers, of course, there they are testing whether you really know, whether you've studied and really know more about the topic. So, for example, if you look at question 2, that question 2 says, give 2 target cells in the human body whose ability to respond to insulin can decrease. Now, you're not going to find that answer in the extract, but of course, we know that we've studied that. So the target cells, we remember your liver cells and your muscle cells are the target cells for insulin, because the liver converts glucose to glycogen, which it stores, and the muscle cells also are stimulated to absorb glucose. So this answer is not in the extract, but you know that you've studied it. All right, and the same with this question over here. It says, name one body fluid that can be used to test for the presence of excess glucose in the body. Again, it's not in the extract, but it's testing your understanding of the word. Like I said, like you can see over here, this is knowledge that you should have, and the answer there is blood or urine. All right, then, section A is going to be 50 marks. Then section B is going to be 2 questions of 40 marks, but they are divided into smaller sections that can be 10 or 12 marks or so, and they will include maybe drawing a table or drawing a graph or a little question with an article from a journal like the previous question that I gave you. But the last section C, is where you need to now write an essay. And unfortunately, many people lose marks in the essay, which is a pity because the essay, as you can see, is 20 marks. All right? So the essay question, 17 marks are given for content and 3 marks for synthesis. So I will explain what the 3 marks for synthesis is in a minute. Now, like when you answer the question, you need to plan it. So you can just take some rough paper and decide how you're going to approach your essay. So this question, which is really, really a nice question, if I was a matric, I would be pretty thrilled to get an essay question like this, because all of this comes from one chapter, and often in an essay question, they can, of course, ask you on different chapters. So you need to get your knowledge from different areas of the life science syllabus. So this is a really nice question because it says, describe the location, structure, and functions of the DNA molecule and the process whereby copies of this molecule are made. So location, that means where do we find the DNA molecule? Structure, so you're going to write a little bit about the double helix and about the nucleotides which make up the DNA molecule and so on. Then functions over here. So we know that DNA is a hugely important molecule because basically it carries all the hereditary material. So everything about you, what enzymes you produce, whether you're tall or short, what the color is of your eyes, etc. And of course it controls the synthesis of all your proteins. Then it says you also need to explain the process whereby copies of this molecule are made. So then we need to explain the process of replication. So replication occurs just before cell division, during mitosis and meiosis, and this is when the DNA molecule makes a copy of itself. So this is a very, very nice question. So you're going to plan it. And, well, I've already explained all of this over here. So I'm not, again, going to go over the whole essay. So you're going to have a little draft. But now when you present your final answer, very important, it cannot be in the flow, in the form of a flow diagram. When you're doing your planning, you can do little arrows and so on, that's fine. But not when you write your final essay. In your final essay, it's going to look something like this. All right, you're going to do it in separate paragraphs, and you're going to do it in a logical way. So you look at your question, and they first wanted to know about the location. So in your first paragraph, you're going to explain where is DNA found. So it's found in the nucleus, or you could say in the chromosomes, and you also get your mitochondrial DNA. So in my first paragraph, I've explained where it is. Now, I just want to go back quickly, but when you write an essay for life sciences, you don't have to do not include a lengthy introduction whatsoever, all right? So when I studied the DNA molecule, I learned this, and that. Do not do that. You get straight to the point, and you just stick to what they are. So we're not in an English essay. You might have a little bit of an introduction and so on. This is not necessary. You just stick to the information that is required of one, all right? Then in the next paragraph, you're going to write all about the structure. So there we go. I'm not going to go over all of this, but there we've spoken about the structure. Then we go on to the functions of DNA. So you put that in the next little paragraph. And then lastly, you explain replication. Now, if you do it like this, then you have presented your essay in a logical way. And you will get, when it gets to the synthesis, you will get 1 mark for presenting your work logically. Now, you will also see that there are lots of little marks over here on the side. So this means that you don't just, so there will be more than 17 facts over here, right? So these are all the possible answers that are correct. So you can say it's a nucleic acid, that it's double-stranded, it's in the form of a double helix, it's made up of nucleotides. So all of these are separate marks that you can receive. So there are more than 17 possible marks. All right? Then just to carry on, so I've already spoken about the logical sequence. So you're doing it in that order. Now relevance is also important. So if in your essay where you now have to describe everything about DNA, you start talking about RNA and messenger RNA and transfer RNA and protein synthesis, that is not relevant to this particular question whatsoever. So you are going to be losing that mark. So relevance means that you only stick to whatever is required of you. Comprehensive means that you have included something on each aspect, and normally there are a minimum number of marks that are required. So you need to include something on the location, something on the structure, or quite a few facts you need to include, something on the functions, and quite a few facts on replication. So comprehensive means that you have included information from each of those sections. So over here you can see if you've got at least 8 correct points for location, structure, functions, and 5 correct points for replication, only then will you get the mark for comprehensive. All right, so with the essays, please bear all of this in mind. Right, now I've been speaking for quite some time. So I've now completed what I wanted to do on how to approach the exam paper. So I've explained to you how to do the multiple choice questions and how you have to choose the correct answer when you are given a statement. I've told you a little bit about drawing graphs. I've told you a little bit about drawing essays. Not drawing essays, about writing an essay. And then I have also told you how important it is to know beforehand that exactly what to study for paper 1 and exactly what to study for paper 2. And then I've also told you that you need to refer to those examination guidelines all the time so that you learn the correct information. So that's what I've been doing now. I'm looking at the time. I've spent about an hour doing that. So I hope that this has helped you. So now we are going to go on to the section on DNA. So this is now on, I'm just having a look in my notes. It is, let me just have a look here. We've done all of that. So we are now on page 12 in the notes. So I hope you've all got that. Okay, I'm going to show you a short little video on protein synthesis because that can be a little bit confusing. I often show my learners videos that have been made by the Amoeba sisters. They are, they're done in a cartoon-like style, but scientifically they are correct and easy to understand. So I'm going to show you one on protein synthesis in a minute. Before we do that, let us just have a look at this on page 12. So there we've got the DNA molecule. Now we're going to answer this question together. So before we even look at the questions, let's see what we've got over here. So we know that DNA is, like I told you, is such a super important molecule because it controls protein synthesis, and you know that in your body alone, all your enzymes are proteins, many of your hormones are proteins, bone, muscles, skin, and so on is all made up of protein. And then, so it carries all your hereditary information as well. So this is now obviously just a little segment of a DNA molecule because a DNA molecule is a very big molecule and it is twisted in the form of a double helix. So I'm sure you all recognize this little picture over here. So over here, these are your sugar portions. And an easy way of remembering that is because your sugars, ribose and deoxyribose, they have ketose sugars. So they have 5 carbon atoms, and this is a little pentagon, isn't it? It's got 5 little sides. So that is your sugar. Please, while I'm talking, obviously the DNA molecule in your exams may not look exactly like this, right? It can be drawn in different ways. The little circle we know is our phosphate portion, and then these are your nitrogenous bases over here, and the little lines are the hydrogen bonds. I'm just going to quickly, I see the phone is ringing, just please ignore that. Oh no, one of my children's going to switch that off. Sorry about that. So these are your little hydrogen bonds over here between your nitrogenous bases. Okay, so let's look at this question. So provide labels for 1. Now, some of you will look at this question and think, oh, no, how on earth do I know what 1 is? But remember, we are testing your understanding. So we look at this one and we say, oh, yes, but this one's it's got exactly the same shape as that one. And this one is joined to, this stands for thymine, of course, and the G stands for guanine. I may be pronouncing it differently to what your teacher pronounces it, but it doesn't matter how you pronounce it as long as you spell it correctly. Now, we know that thymine always bonds to adenine and guanine always bonds with cytosine. So this one over here is adenine, so obviously number 1 must be adenine as well, isn't that correct? Then the next question says, provide labels for number 2. Well, I've already told you that this is the sugar deoxyribose. There we go. And then they want the label for number 3. I've also told you that these are your weak hydrogen bonds between your nitrogenous bases. Then it says, give the number of nucleotides in the diagram. Now, a nucleotide is a building block of a nucleic acid. All right, we also sometimes say a nucleotide is a monomer. Monomer is another word for a building block. So a nucleotide is a building block of a nucleic acid. But we also know that nucleotides are made up of 3 sections. So over here, the sections would be over here, your sugar, right? Sorry, not your sugar, that's your phosphate, your sugar, and your nitrogenous base. So that over here is 1 nucleotide, right? Because it's made up, like I said, the nitrogenous base, the phosphate, and the sugar. So in this molecule, there will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nucleotides. All right? Then the next question says, give 1 difference between the nitrogenous bases found in DNA and those found in RNA molecules. Now, we're going to be talking about RNA when we look at the little video on protein synthesis. So you will see they are going to talk about the differences once again. Now, we know that in DNA we have the sugar, deoxyribose, whereas in RNA we've got the sugar ribose. So I'm not reading the question properly myself. There I've made a mistake because they've actually asked for the difference between the nitrogenous bases. So I haven't read this question correctly. So once again, you need to read it correctly. So we know that that RNA has got uracil instead of thymine, right? There we go. There's uracil replaces thymine in the RNA molecule. Right. Then it says there are 2 processes involved in protein synthesis, namely transcription and translation. And there in your learner resource, they've got a very nice explanation of transcription and translation, and it is also in your examination guideline, and you obviously need to know this very well. I'm not going to go over it because it's going to be in the little video that I'm going to show you. Then You mustn't get confused with DNA replication. Now, unfortunately, I haven't got time to go over DNA replication as well, but this is basically when the DNA molecule makes a copy of itself, and it makes a copy of itself just before mitosis and just before meiosis, and of course it occurs in the interphase of the cell cycle, so just before mitosis and meiosis. And this is again nicely explained in Mind the Gap and so on, and it's also in the examination guidelines, so you need to know this very, very well. Okay, so before we look at the questions on protein synthesis, I am going to quickly show you the video clip on protein synthesis. Let me have a look. Okay, I had it on my screen earlier on. Just give me a little bit of time till I find that. Just be patient, Matrix. There we go. Actually, I'm just going to go out of this first because I've just remembered I need to switch on the audio first, which I haven't done. Oh, dearie me, let me just find where I know I did it the last time. Okay, just admit somebody over there. Let's just try. Okay, and then open share tray. Right, I do hope that this is playing.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid. leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious in a trait. Let's take an example.
Okay, let me just have a look. Okay, so now we're going to do some answers in your tutoring resource. But what I want you to do is to work in little groups, and your teacher can even help you, and then we're going to be going over those answers. So the first one I want you to look at is question 2 on page 14. All right, if you can look at question 2 on page 14, and I will also put it on the screen. And then when you finish that, you can do question, let me just have a look what I thought, maybe do question 4. Okay, so if you can do question 2 on page 14, and then do question 4. I'm going to give you a little bit of time, and then we can work through the answers together. I'm going to put it on the screen as well.
Mr. Clerk, you'll notice I'm removed the one school out of the meeting. It seems there's no teacher there, so they'll have to get back in. I don't know if you noticed.
I just want to confirm with you, Mr. Farmer, that you can all see question 2 in front of you on the screen.
No, I don't see it. Can you maybe just try share it again, please? I don't see it on the screen.
Oh, you're just looking at me at the moment.
That's right, yes.
Yeah, okay, that's not right. Okay, let's just go out of here. Okay, let me just have a look. So, open share tray. Let's have a look. Protein synthesis. Desktop. And Right, are you able to see this now on the screen? The questions, Mr. Farmer?
I can see it clearly now, yes, thank you.
I'll just ask the pupils in groups to work through question 2 and 4, and then we're going to be going over that.
That is good, yes, that's fine. If you look at the total of the question, then give him more or less the same time of amount of time. You know, if it's 5 to 10 minutes, then give him more or less that time. Yes. Okay, it's going well. I trust the learners are also following well. I'm happy with what I'm. Thank you.
Hello, William. Hello, sir. Hello, ma'am.
Wil jy met meneer Farmer praat? Hello?
What's that? I didn't hear the question. What is the question?
Yeah, I wanted you to scroll down a little bit. I just want to see the diagram. No, I mean up. Sorry.
Sorry, okay, that's absolutely fine. So this is the diagram over here.
Yes. Okay.
Okay, you can just tell me when you want me to scroll down.
No, I think it's fine.
Okay. Okay. Okay.
Thank you.
Okay.
Mr. Clerk?
Mr. Farmer?
There's a request from one of my teachers that if they can also have the video that you showed with them, the one on protein synthesis. If perhaps if it's too big, you can tell me where's the link, the YouTube link, then I can download it and share it with my teachers.
Mr. Farmer, I use it's amoeba sisters and then protein synthesis. And in fact, all their videos are very good. So you will find ones on replication, they're on monohybrid cross, dihybrid cross, and so on. So I use them quite a lot with my matrix, and I really, really like them. I think they're very good.
Okay, can you just maybe tell me on WhatsApp again, because I missed part of it, and then I will check it. Okay.
Okay.
Thank you.
Matrix, I hate to do this because I would really prefer you to work out all these answers on your own. I think not all the learners have got this learner resource, so perhaps for next time the teachers can get hold of Mr. Farmer and organize it. All the schools have got this, but I would like to try to at least finish the section on DNA in this tutoring session. So I really feel bad that I'm going to cut you out, but you will have access to these questions, and I will be able to go over some of these answers with you at a later stage. So let's just see how you're doing so far, Matrix. So yeah, we've got a diagram. Now let's just have a look. So over here we've got the ribosome, and there we've got this obviously is transfer RNA, and here we've got some amino acids. Here's another transfer RNA molecule, and this obviously is messenger RNA. And this messenger RNA has been built up opposite one of the DNA strands in the nucleus of the cell. Mr. Do you want to say something?
Oh yes, we've got the picture of the diagram. We've got the diagram. Now we're answering the question. Thank you.
Okay. Do you mind if I go over some of the answers? As I said, I'd love to give you more time to do this together with your pupils, but I thought just to save some time so that we can cover some more work, I'm going to go over some of these questions now, and then the pupils or the learners can do the others in class, and we can then maybe put up the answers onto MS Teams at a later stage. Is that fine with everybody? I hope it's fine.
It's fine with us, ma'am. Thank you.
Okay.
Good idea, yes.
It says identify the stage of protein synthesis that is shown in the diagram. So remember the two stages are, as we saw in the video, transcription and translation. So transcription is when the messenger RNA is built up opposite one of the DNA strands. So this over here must be translation. Then it says identify molecules X and Y. So this molecule over here is a transfer RNA molecule. So X is a transfer RNA molecule. And over here, Y over here is then the messenger RNA molecule. Then it says state the term for the group of 3 nitrogenous bases indicated by V. Now, this is V over here. Now, on your DNA molecule, your nitrogenous bases are arranged in groups of 3, and we call those base triplets. And then on the messenger RNA, those same base triplets are called codons. And then on the transfer RNA, those same base triplets are called anticodons. So the answer for V is an anticodon, right? This is also nicely explained in Mind the Gap. Then it says, give the nitrogenous bases on the DNA strand that code for the bases UAU on molecule Y. So let's find molecule Y. So here we've got UAU. But now remember, you are going to be working backwards, right? Now, what you've got to remember, firstly, that RNA has got uracil instead of thymine, right? And this UAU, this codon was built up opposite a base triplet on the DNA molecule. So now we are working backwards. So on the DNA molecule, opposite U, which now you will obviously find adenine, because remember, the messenger RNA doesn't have thymine, it's got uracil in its place. So opposite U, you would find adenine. opposite adenine, you would find thymine, and opposite uracil, you would once again find adenine. So your answer there then is ACA. And then the next question is, use the table below to identify amino acid W. Now, firstly, I do hope that everybody got those first 4 questions right. And if you got those first 4 questions right, then you are doing great. If not, then do go over the section on transcription and translation again in your textbooks or in Mind the Gap. The important thing to remember here is that the messenger RNA and transfer RNA do not have the nitrogenous base thymine. They've got uracil in its place. All right? And you all know that messenger RNA is built up opposite one of the DNA strands, and it then moves into the cytoplasm, and then the anticodons of the transfer RNA are going to join to the correct codons on the messenger RNA. Right. Now the next question, now I'm going to let you just look at that for a second, because I think some of the schools do not have this learner resource. So hopefully you will have it in front of you the next time, because this is what I've been using for all of this lesson. So this question says, use the table to identify amino acid W. All right, let's quickly have a look where amino acid W is. All right, there it is. All right, so it is the amino acid that is going to be joining onto this part of the messenger RNA molecule. All right, these are the names of the amino acids, and there is the anticodon on the transfer RNA molecule. Now, again, you've got to look at this carefully to work out the correct answer. Maybe I should just okay, let me just go over this with you. I think that's going to save a little bit of time. Right, so firstly, remember we've worked out what, so this is messenger RNA, UAU. So what would, and this is our codon, so what would the anticodon be of the transfer RNA molecule that is going to join up over here? So again, opposite U, you're going to have adenine. So it's going to be adenine, and then over here it's going to be not thymine, but uracil. So it's going to be AUA. So we go back to the table, AUA. That is the anticodon of transfer RNA, and the amino acid that's going to be joining up there is tyrosine. I hope you all got that one correct. As I said, this applies mainly to the people that have got the notes, because the other people are only seeing the question for the first time now. So obviously that becomes a little bit more difficult, because you've got to have time to look at the questions carefully. Now the next question says, name and describe the process that occurs in the nucleus to produce molecule Y. So to produce molecule Y is this messenger RNA. Now to produce that molecule, we go through the process of transcription. So you've now got to explain that whole process of transcription, which we did earlier on. I think here it is. There we go. I'm not going to read through all of that, but there you need to write down all those little points and describe the process of transcription. Okay, now you are going to do for next time or sometime during the week, you can do question 3, which is very similar to the question that I've just done with you, because there you can again see a ribosome, messenger RNA, transfer RNA, and amino acids. So you can do this one at home, and we will give you the answers later on to see whether you understand this. Now we're going to go on to question 4, which I also asked you to do over here. And again, let's look at the question first. So it says a section of a DNA molecule has the following base sequence: CTTACA. Name the nitrogenous base represented by C in the DNA molecule. Well, that's a nice, easy question because we know that C stands for cytosine. Now, here's a little bit more difficult, this question. It says the percentage of guanine in this DNA molecule is 30%. Give the percentage of thymine in the same molecule. Now, remember, guanine always joins up with cytosine. Those two always bond together. So guanine and cytosine together then are going to make up 60% of the DNA molecule. Am I not correct? So then we are left with 40%. So 40% has to be shared between thymine and adenine because we know that thymine and adenine always join together. So that means that thymine will then make up 20% of that same molecule. Can you see? We had to do a few little calculations, simple calculations, to work out the percentage of thymine in the same molecule. I hope everybody understands that, and if you understand that, then you're doing fantastically. Then it says, give the messenger RNA sequence from left to right for the segment of DNA. So let's just go back to this again. Now this is a section of DNA. Right. So opposite C, we're going to have a G because we know those two join together. Opposite T, now many of you are going to say, oh, we're going to have an A. But, opposite T, we would have A. That is correct. Then opposite T, we have A. Then opposite A, we now would want to say, okay, adenine and thymine bond together, so we'd be tempted to write down T. But of course, we've got to remember that RNA doesn't have a T, it's got uracil instead. So opposite A, you're going to have uracil, a U. Opposite C, you're going to have a G, which is guanine. And opposite A, again, you're not going to have thymine, you're going to have uracil. So the answer then would be G, A, U, G, U. Okay. Then again, it says the table below shows the DNA triplets that code for some amino acids. All right. Here's the DNA triplet. There's the name of the amino acid. Now, you don't have to ever learn the names of these amino acids, all right? Then it says, give the sequence of amino acids that would be coded for the section of DNA above. So you've got to look at the DNA sequence above. So the first three are CTT, and then we go back to the table. Right, CTT, the name of the amino acid would be glutamic acid, and you write that down. Then the next three base triplets are ACA. So we look at the table, there's ACA. So the name of the amino acid will be cysteine, and you write cysteine down. Then we go, it says, give the anticodon for the amino acid, I don't know how you pronounce it, I'm just going to pronounce it methionine. So the anticodon, remember, is going to be found on the transfer RNA molecule. So now we again have to work backwards again a little bit. So there we can see this is the base triplet for methionine. Over here it is, sorry, on the DNA molecule. So it's best if you write this down, and I'm also going to write it down so that I don't get confused. So we want to know the anticodon for methionine, which is the last one in the table. So again, we're going to work backwards. So if TAC is the base triplet on DNA, then on messenger RNA, then we know that opposite T, you're going to have an A, and opposite A, you're going to have a U, and opposite C, we're going to have a G. So that is now the codon on messenger RNA. I hope you're writing that down. So we've got AUG. But now remember, we don't want to know the codon, we want to know the anticodon. So let's look at the anticodon. So opposite adenine, you're going to have not thymine, you're going to have uracil. Opposite uracil, this is now the second nitrogenous base of the codon on messenger RNA. So opposite uracil, you're going to have an adenine, and then opposite guanine, you're going to have cytosine. So the answer there, I hope I'm not working too quickly, it should be UAG. Maybe I should look at my answer book and see that I'm correct, that I haven't done something wrong. I think I have done something wrong because it says UAC. Okay, then I worked too quickly, but I'm sure your teacher, I can see what the mistake is. So in my last nitrogenous base on the codon was a G, so opposite that you obviously have a C, which is cytosine. All right, I'm sure all your teachers will anyway be able to explain this all quite easily. Then describe how a mutation on DNA may change the structure of a protein. Now, very often, you need to look at words and marks. So over here, it says describe. So for describe, you've got to write down a whole process, all right? Also, it's for 4 marks, so you have to give a little bit of detail. If you look at the previous question, it just says give. So give just means give the answer briefly. You don't have to explain it. But describe, you do need to explain it. Now we know, obviously, a mutation on DNA, how will it change the structure of a protein? So first of all, obviously, the DNA code will change. And then the messenger RNA that is formed opposite the DNA will also have different codons, all right? And this will mean that different transfer RNA molecules are going to join up with those codons on the messenger RNA molecule, and now you're going to have completely different amino acids joining together, and you're going to have a completely different protein, right? So you're describing it. This word tabulate is often used in exams as well, and that means draw a table. So a table with rows and columns and headings and so on. So tabulate 2 structural differences between a monomer of RNA and a monomer of DNA. Now remember, the monomers are your nucleotides, and we know, so in your table you're going to have one heading RNA and another heading DNA, and then we know that the sugar in RNA is ribose, whereas the sugar in DNA is deoxyribose. And then we also know that DNA has got the nitrogenous bases adenine, thymine, cytosine, and guanine, whereas in RNA it's got the nitrogenous bases adenine, uracil, adenine, uracil, cytosine, and guanine. So there we've tabulated the differences. You'll see it's 5 marks, so you also get a mark for drawing the table. Now I need to look at my time because you also need to do the online test. Now, the little pre-test. The questions are difficult and maybe you haven't studied all this work, so do the very best you can. You should all have the pre-test with you, at least the teachers should have it with you, but for those of you that don't have it, I'm going to put it on the screen, and you are now going to have the rest of the 15 minutes to answer those questions questions. They're all one-word answers, but you do have to think of them, the answers carefully. Now, unfortunately, I just want to show you the next question. The next question is about these DNA profiles, and there are 2 questions on DNA profiles. And you hopefully will have time to go over this in class with your teachers. So these DNA profiles are very important in paternity cases where you want to see who the father is of a child. They're also often used in crime scenes, and each person has their own unique DNA profile. And basically what we do with these DNA profiles, as you can see here, you can see there's blood from broken glass, and we want to see whether person P, Q, and R was maybe present at this crime scene. I haven't read the whole question, but it's probably a crime scene. So you've got to look at these bands of DNA and see which ones match the best. And just looking at this quickly, I can see that in Q, the second band matches over here, the third band matches, the fourth band matches, the fifth and the sixth band. So more than likely, Q was present at this crime scene, and because Q has the most bands matching the DNA profile over here. So this is something that I also want you to go over in class, and unfortunately I didn't get onto meiosis, so I will do that in the next question, but now I'm going to leave this section and let you do the answers to the little pre-test, which I will also then put on the screen now. Okay. Okay, it says 1 hour, but you don't need that time. I just want to confirm with Mr. Farmer that especially for the people that do not have this in front of them, that you can all see what is on the screen. So the first one says, for example, you need to write down the biological term, and the first description is 2 alternative forms of a gene at the same locus. Now remember, this is in the section on genetics, so if you haven't studied this yet, you might not remember this, but do try. You've done a year of life sciences now, so do try and write down the correct answer. Mr. Farmer, can everybody see this, the test?
I can see it, Mr. Clerk. I'm sure everybody else will see it. Thank you.
Yeah. Then one of the teachers can maybe just say when they would like me to scroll down.
Mr. Clerk, I think the teachers or the learners might have the hard copy in front of them, so they should be able to do it on their own now.
So if any of the teachers don't have it, then they can just let me know, and then I will scroll down as soon as the pupils have finished with these 8 answers. Okay, grade 12, you can see that quite a few of these questions were on genetics and meiosis, which we didn't discuss today, but some of them I hope you would have recognized from the work we did today on DNA. You can see the last question, those are typical questions that you would get in the exam paper. And of course, the terminology is very, very important in life sciences as well. Now, I'm sure it will be absolutely fine if you quickly finish these questions first before you go home, because I see it is now 1:30 already. And do remember that I think it is absolutely fantastic that you and your teachers are here on a Saturday doing this work. But remember now to use your time between now and your mock matric and your end of the year exams, which are coming up so very, very quickly, to really do a lot of revision at home. All right? At the moment, we're not allowed to socialize with people anyway because of this COVID virus. So it is important so you may as well stay at home and do a lot of studying. You will be happy in the end that you've put in all those hours, and it really is hours. And I don't know if Mr. Farmer wants to say anything. I'm happy to still sit here and answer questions. And otherwise, I will see you in 2 weeks' time. And I do hope that many of you, that you tell your friends to come along as well. And I hope that it has helped you as well. But I'm first going to ask Mr. Farmer if he wants to say anything.
Yeah, thank you, Mr. Clerk. I really appreciate it. I first want to say thank you to you. I was following your lesson toggle between Afrikaans and the English sessions, and I'm very pleased with the interaction. Interaction from your side, obviously learners can't interact, but I would just want to echo what you said, that this is the time for our matrics to focus. I always say the celebrations can wait. Now you have to put in the hard work, and I'm also very pleased and proud to see you guys here on a Saturday morning, Saturday afternoon. It shows your dedication. Just persevere, do your best, and you will reap the fruits of your labor eventually. So I just want to thank you, Mr. Clerk, and I want to tell you next in 2 weeks' time, your session will be on again. And just make sure that your teachers take in the test that you just wrote now, and then you can ask your teacher to mark it, or hand it out in class on Monday and get you guys to mark it among yourself. But this is just a pre-test, learners, to see what your understanding is of the work. And then at the end of the tutoring session in October somewhere, we will give you another test to see what the impact is of the tutoring. All right. So I want to wish you all a very good weekend. Take time to break, to relax, and then do some work as well this weekend. Don't take the whole weekend off. Okay, with that, I will stop the recording now, Mr. Clerk. If you want to, you can maybe stay on a few minutes, just in case there are a few questions. And then I'll see you again in 2 weeks' time. Thank you very much, everybody.
Thank you. Okay, so I'm more than happy to answer any questions. And otherwise, as I said, I will then see you in 2 weeks' time. Does anyone have any questions? Okay, then everybody enjoy your weekend and work hard, obviously. That's super, super important. Work hard and